Hi, Miss Nikki here. Welcome to Chapter 2. You can see from the title we're going to talk about chemistry and how chemistry leads to energy. Don't be too afraid. We're just going to talk about the basics of chemistry because we have to talk about some more complex items later on in the semester, and you need to have a foundation or a basic understanding of chemistry. So let's get right into talking about chemistry. So the two major categories that we're going to talk about are called basic chemistry and biochemistry. We've talked just a little bit in Chapter 1 about some of these different systems, like the nervous system, the digestive system. We talked about the cardiovascular. So the cardiovascular helps us pump the heart. So chemistry is the underlying principle that guides all of these physiological reactions within the body. So first let's look at basic chemistry. Basic chemistry starts with basic definitions. There's matter, there's energy, and then we're going to break it down into types of energy. So matter is anything that occupies space or has mass. So that would be a human. I have mass, the desk has mass, your textbook has mass, the car has mass. It can also be a liquid like water. Solids, I just gave you some examples, and then also some gases. Your book also includes plasma in here, but I'm not going to include it. We'll just stick with the three basic forms of matter. For energy, energy is what we need to do work, to put matter in motion. So in order for me to move my arm or to move my mouth to make this video, I need energy to get these muscles to work and carry out the task that I'm asking it to do. So the two types of energy here, we have kinetic and we have potential. So kinetic is energy in action. You could think of kinesiology. So kinesiology is the study of the body in motion. So kinetic energy is when you're putting energy in. So there's two examples here. If you're riding a bike up a hill, you're putting in lots of energy. You're moving your legs, you're putting energy in. When you get to the top of the hill, that's when you have a lot of potential energy. And then all you have to do is let go. You're not putting any more energy in, and you go flying down the hill because of the energy that you put in the beginning. Same thing with this picture here. They're showing somebody diving off of a cliff. So to walk up the stairs, you're creating potential energy. You're using kinetic energy, you're creating potential energy when you're here at the landing, and then when you jump off, you're releasing that kinetic energy that you built up. So think of kinetic as action and think of potential as stored. The last slide, we talked about two general types of energy. We talked about potential and we talked about kinetic. We can break down those two categories and talk about very specific forms of energy. We have chemical, we have electrical, and we have mechanical. So chemical energy is stored in the bonds of molecules. So we've seen glucose. This is the molecular formula for glucose. And what we're saying is in the bonds of carbon, there's 6 carbons, there's 12 hydrogens, and 6 oxygens that make up glucose. And in these bonds, we have energy. And that energy is stored energy. So, but it's also chemical energy. So chemical energy would be the same thing as potential energy. Then we have electrical energy. Electrical energy results from moving particles. So this picture or figure at the bottom here, we can see a charged molecule moving through these two complexes, and as that charged particle moves, it creates enough energy to turn ADP plus P into ATP. We're just changing from an electrical energy into a stored energy. This can also be called chemical energy. Mechanical energy is directly involved in moving. What other general form of energy was directly involved in moving? That was kinetic, right? So kinetic energy is the kind of overall or general type of energy, but we're specifically talking about mechanical energy. And maybe this picture over here will help when we look at all three of these energies, chemical, electrical, and mechanical. So here's the sun. The sun is shining light onto the tree here. And this type of energy is electrical. If I can spell, right? Electrical. I got too happy there. All right, so the sun shining onto the tree, we're converting electrical energy that's in the sunlight, we're turning that into chemical energy. The tree is going to store glucose. Then the moose here is going to eat the leaves, it's going to eat the glucose, and it's going to turn the glucose to mechanical energy. So we have all three forms of energy that are described here in this one figure. So even though I've talked about energy conversions, this slide here is just stating that energy can be converted from one form to another. We've just seen how electrical was converted into chemical, which was then turned into mechanical energy. And I've given some examples here, electrical to maybe to light or to thermal. So this is the socket, electrical socket in your house, and you're using it to power a lamp or a heater. You can also take a chemical like coal and you can use it to power a train. So energy conversion is inefficient though, and some energy is always lost as heat. One of the best examples, there's two here. One that we know because we probably drove to the store the other day. We have gasoline molecules, so they're saying in these chemical bonds that are holding this gasoline molecule together, this is chemical energy. It's also stored energy, so we're going to call it potential. And then the oxygen and the gas go into the combustion engine. I'm not talking about electric cars, right? And this causes the wheels, the axle to move the wheels, and then the car moves forward. You're giving off heat in the process. So when you turn the energy or convert the energy from chemical into kinetic, and this is particularly mechanical, right? We're causing something to move. When you change from chemical energy to mechanical energy, you're going to give off some heat. And we know this because we put our hand on the hood and it's warm. This is an example we've seen already, cellular respiration. We take food in humans, particularly glucose, sometimes fructose, and we take oxygen and we put it into our cells. This is a cell. It undergoes cellular respiration and it takes the energy that's in the bonds of the glucose and stores the energy in something called ATP. So ATP is also chemical energy. But then we're going to take chemical energy stored in ATP and we're going to use it to do work in the body, which we would call mechanical. So in changing from one form, glucose, into another form and then using it to do work, we're going to release heat in the process. And this is great for us because we want to maintain our body temperature at 89.6. So the heat that's given off by cells helps contribute to our overall body temperature homeostasis. So we looked at matter, and we looked at how we move the matter. So if we want to get the matter to move, we have to use energy, different types of energy. Now we're going a little deeper into what is matter. Well, matter is made up of elements. Elements cannot be broken down into simpler substances. So if something takes up matter, let's say the example I gave you earlier where it's carbons and hydrogens and oxygens, the elements that are in glucose are carbon, hydrogen, and oxygen. So four elements that make up 96% of the body are carbon, oxygen, hydrogen, and nitrogen. You could look at a periodic table and you could see all the elements that we've found so far. Atoms would be the next layer in. So we are here at matter, then we look at elements. Inside elements, you're going to have atoms. So atoms are the building blocks for each element, and they give that element its specific physical and chemical properties. And basically, it's the smallest particle of an element with the properties of an element. Usually we'll use atomic symbols. This is what you see on the periodic table. Usually it's one or two letter combinations if we want to do shorthand. So this one we probably have seen already. Carbon, oxygen. Some of these other ones, this is calcium. This is iron. Phosphorus. Iodide. This one is potassium. And this one is sodium. And these are ones that we'll see frequently throughout anatomy. So now we're looking at subatomic particles. So subatomic means inside the atom. So now we have matter, and then we're looking inside at elements, and then we're looking farther inside an element, we're looking at atoms, and then when we get inside the atom, we have subatomic particles. Those subatomic particles are called protons, neutrons, and electrons. So there are 3 subatomic particles: protons, neutrons, and electrons. And you can see that proteins are positive, electrons are negative, and neutrons are neutral or have no charge at all. This figure shows you the atomic structure, giving you some different elements. So this is hydrogen element, this is the helium, and this is lithium. They're telling you the number of protons, neutrons, and electrons that each one has, and then they're showing you in a figure what it would look like. So these out here, these are the electrons, as the legend says. These are the protons, so these are positive. The electrons are negative, and then the neutrons are neutral. So I want you to see how the electrons are kind of orbiting around the core, and the core is containing the neutrons and the protons. This slide is basically just trying to get us to understand some basic definitions between what's a molecule and what's a compound. So if I take some atoms and I put those atoms together, they're going to form molecules and compounds. Molecule is any 2 atoms, and a compound, it has to be different. So 2 oxygens together would be a molecule, but 2 oxygens together would not be a compound. The next slide that we have to look at talks about the difference between a solute, a solvent, and a solution. So we have solvent 1, solute 2, solution 3. These are 3 terms you need to know. So the item that's doing the dissolving is the solvent. In the body, it's usually water. The solute is the item being dissolved. So let me give you an example. Let's say we have salt. So this is sodium and chlorine, and we put them together, and we have salt. So let's say we take the solute, which is salt, and I dissolve it in water. The resulting solution would be salt water. We can also do something a little bit more fun. So let's say the solute is chocolate, the dissolving item is hot milk. Your resulting solution is going to be hot chocolate, right? On to everybody's favorite topic, which is chemical bonds. You can see from the definition here, chemical bonds are energy relationships between the electrons of the reacting atoms. So remember, the electrons are the negatively charged particles. We call them subatomic. And they're kind of in that cloud around the protons and the neutrons that are in the core. The 3 types of bonds that we're going to look at are called ionic bonds, covalent bonds, and hydrogen bonds. So in ionic bonds, we're going to talk about atoms that gain or lose electrons. Some people call ionic bonds the transfer of electrons. Covalent bonds is when we're going to see a sharing of electrons. So sharing of electrons. And a hydrogen bond is where we're going to see attractive forces between one side that's positive and one side that's negative. So here's a great example of ionic bonds. We're going to look at sodium and chlorine. This would be salt. And remember that ionic bonds we're going to transfer. What specifically are we going to transfer? Electrons. And what are electrons? They are negative subatomic particles that make up an atom. So here's the sodium. It has 11 protons and it has 11 electrons. So you can count all of these and see that there's 11. But let me explain the ring structure. So in this first ring, you hold 2 electrons. The next ring holds 8 electrons, and the last ring will hold 8 electrons. Do you see how this sodium has 1 lonely electron on its outer ring? Chlorine, on the other hand, is only missing one. It has 7 on the outer ring, and it would love to have one more. So in this instance, when a sodium atom comes in contact with a chlorine atom, the sodium gives up its electron to the chlorine, and now they're both happy. So this outer ring has 8, and this outer ring has 8. And so this is called an ionic bond. You can see here at the bottom some definitions, anion and cation. Some people say anion and cation. Anion, cation, either way is fine. So anions is if you have a negative charge, and a cation is if you're something that's positively charged. So what they're trying to say, let me erase some of this. If the sodium atom had 11 positive protons and it had 11 negative electrons, well, now it gave one of its electrons away, so now it only has 10 electrons, and those are negative. So if I have 11 positive items and I have 10 negative items, I'm still slightly positive. If I'm slightly positive, I'm going to be a cation. For the chlorine, it had 17 positive protons and 17 negative electrons. But what happened? The electron from sodium was given to the chlorine, so now it has 17 positive and it has 18 negative. This makes the overall atom slightly negative. If you're slightly negative, you're an anion. The next bond on our list are covalent bonds. Covalent bonds, if you remember, was sharing of electrons. So instead of transferring, we're going to share electrons, but this is where it gets a little tricky. There's two different types. There's polar and there's non-polar covalent bonds. Polar just means opposite. So one side of the molecule is going to be negative and one's going to be positive. So this is a great example over here of polar. This side is slightly negative and these sides are slightly positive. So we call this a polar molecule. Specifically, this is a polar covalent bond. So there's an unequal sharing of electrons between two atoms that makes one side positive and one side slightly negative. So you can see if we take two oxygen atoms and they react, they're now sharing these electrons in the middle. They didn't transfer any electrons. Both atoms are actively sharing those electrons. But then count up the number of electrons. Over here I've got 6 negative electrons, and on this side I only have 3 electrons. So this middle part here is more negative than the outer part. So this part is slightly more positive. We would call that a polar covalent bond. So the equal sharing of electrons, the unequal sharing of electrons that results in a polar molecule where one side is slightly negative and one side is slightly positive. For nonpolar covalent bonds, this is when we have an equal sharing of electrons between atoms. Basically, you have an electrically balanced molecule. So here are 4 hydrogens, and they're going to react with this carbon atom. And you can see the outer ring of carbon, it only has 4 electrons, but here's 1, 2, 3, and 4. So it's almost like a lock and key. This hydrogen will come over here and share, this one will share here, here, and here. So you get this perfectly symmetrical resulting molecule where there's 2 electrons and 2 and 2 and 2. So you have a paired electrical bond, but it's also nonpolar because it's electrically balanced. The last bond that we need to talk about is called a hydrogen bond. So when we do have polar molecules, what did we say polar was? One side's positive, one side's negative. So these are examples of polar molecules here. So this side is the oxygen and it's slightly negative, and these sides are the hydrogens and they're slightly positive. So what happens with hydrogen bonds, they usually draw them in this dotted line here. Hydrogen bonds is where there's just an interaction. So the negative side of one molecule associates with the positive side of another molecule and forms this somewhat weak interaction. The reason I say somewhat is individually they're weak, but if you put a bunch of hydrogen bonds together, they're quite strong. So this leads us to chemical reactions. If we want to put chemicals together and we want to break them apart, we're going to show that by using a chemical reaction. I've kind of already showed you a chemical reaction. Here's the example at the bottom, cellular respiration. We eat food, which is glucose. We breathe in air, which is oxygen. We convert it to carbon dioxide, water, and ATP. And remember, this is what we're going to use to do work within the body. So this is how you would write the equation. This is glucose, and this is the oxygen, and this is the carbon dioxide, and this is the water, and this is the energy that we're going to get out. So the reactants versus the products. Whatever you put in is the reactant, and whatever you get out is the product. That seems pretty easy, right? So now to the next general topic. Everything we just talked about was basic chemistry. Now we have to talk about biochemistry. So how do chemical composition, how do chemicals and chemical reactions affect living matter? Again, we have two subgroups underneath biochemistry. We can look at organic compounds and we can look at organic compounds. And the basic definition is organic compounds contain carbon. Sometimes they'll also have hydrogen. Inorganic compounds do not contain carbon. So when we talk about inorganic, we're going to talk about waters and salts and acids and bases. When we talk about organic compounds, we're going to talk about carbohydrates, fats, proteins, and nucleic acids. Those all contain carbon. First topic we have here is water, and realize that water makes up the majority of a cell. It is the most important inorganic compound because of these 5 properties. So with a little bit more explanation, we can look at high heat capacity. High heat capacity basically is saying absorbs and releases heat with little temperature difference. Probably the best way to look at that is if you have the sun and it's shining down on a body of water like a lake, the lake is going to absorb the heat from the sun all throughout the day, and then when the sun is gone, it's going to release the heat really slowly. And this is going to leave a more homeostatic or even temperature for the marine life that's in the water. It would kind of be hard to survive if the water was boiling in the afternoon and it was frozen solid at night. High heat of vaporization we've already seen in chapter 1. We talked about sweat, right? So what happens when you sweat? When your body's hot, you're going to release sweat. That sweat is hopefully going to dry, turning from a liquid to a gas. And when we change states from liquid to gas, we're going to take tons of heat away as well. So the whole point of sweating is your body's trying to cool itself down. You sweat, it has water in it. You sweat, that water is changed from a liquid to a gas, and when it does, heat is pulled away from your body. The polar solvent property of water is also very important. So because the majority of cells contain water, water is one of the solvents that is used in the body for chemical reactions. So anytime we want to break things down, put things together, that also falls under reactivity. We haven't talked about hydrolysis and dehydration synthesis yet, but we will. So the fact that water can participate in chemical reactions and is vital for some chemical reactions to occur. And then also you can think about your body's major transport medium, your blood. In your blood you have plasma, and plasma is made up mostly of water. So if you didn't have water, you wouldn't have as much plasma, you wouldn't have as much blood circulating, you'd have really low blood pressure. And then the last one's pretty self-explanatory. You have some cushioning due to water. It protects certain organs from physical trauma. The next topic is salts. Salts are ionic compounds that dissociate into separate ions in water. So we've already seen salt, right? We saw our sodium chloride. What they're saying is if I take this sodium chloride and I put it in water, I can pull apart those individual ions. Remember, the sodium gave up an electron, so it became more positive, and the chlorine took on an electron, so it became negative. So anytime you see something with a plus or a minus after it, we just call these ions. You might also see the term electrolyte. Either one is fine. And these are going to play roles, specialized functional roles in physiology within the body. So we're going to see all 3 of these are very important in muscle contraction. So you cannot have muscle contraction if you don't have the proper amount of these ions. Iron, this is in your blood and helps you carry oxygen. We'll talk about that more in 202. This figure is trying to show you how we move some of these ions. Remember, these are ions or electrolytes. How we can shuttle these inside and outside of the cell. And it'll become important later on in 201. How do I know this is outside the cell? It says extracellular. We'll talk more about what cytoplasm is, but this is inside of the cell. So we have a lot of ions inside and outside of the cell, and they're going to move back and forth. And this is actually really important with nervous cell transmission and muscle contraction. So here is my who cares slides, right? Anytime you see this clinical or homeostatic imbalance slide with the orange heading, I'm basically just trying to point out some key topics to you as to why this is important. So ionic balance is vital for homeostasis. If I said you need sodium and you need potassium and you need calcium in order for your muscles to work, when I say muscles, I'm talking about your heart, right? Skeletal muscles, muscles that are on your bones, your heart, any of your smooth muscles. All of these ions, the balance of them is necessary for your body functions. The kidneys, which we won't talk about till 202, but they play a role in maintaining the balance of electrolytes. So remember, the kidneys decide what you're going to keep and what you're going to get rid of, and whatever you want to get rid of goes out in your urine, and whatever you want to keep goes back into your bloodstream. So your kidneys play a really important role in making sure that the amount or the balance of these ions is kept in proper order. If electrolyte balance is disturbed, you can have all organ systems ceasing to function. So many of them require several different ions in order to function properly. Acids and bases, next favorite topic. So acids and bases are both electrolytes. They ionize and dissociate in water. So what they're saying is if we have a compound and we put it in water, it will pull apart into ions. That's all it's saying. So this is an example of an acid. If I take this and I put it into water, I will have a dissociation and I'll see the ions. So basically water gets in between the hydrogen and the chlorine and pulls it apart. This is an example of a base. A base is a molecule. Water is going to break it apart into ions, but you're going to have this particular ion released. So when you see something that's a base, what we're saying is if I take that substance and I put it into water, it's going to release lots of these hydroxide ions. If I'm an acid and I put the substance in water, I'm going to see lots of hydrogen ions. So acids dissociate into hydrogen ions when placed in water. Bases dissociate into hydroxide ions when placed in water. Sometimes you'll hear the word alkaline as well. Either one is fine, alkaline or base. Here's a pH scale. They call it a potential hydrogen scale, basically just to show you different substances and how they would rate on a pH scale. Are they more basic, more acidic? And so you can see here we have neutral in the middle, and neutral would be blood, water, milk. And then we have pH 14, which means very basic or alkaline. And then we have 0, which means very acidic. So you can see wine, lemon juice, the hydrochloric acid example, black coffee. So if you have an acid solution, you have an acid, you put it into water, you now have a solution, right? So we could go back and say the acid is the solute, the water is the solvent, and the solution would be an acidic solution. So an acidic solution should release lots of H pluses. These are hydrogen protons. An alkaline or basic solution should release lots of OHs, but sometimes they refer to it as very little hydrogen protons because we're really measuring hydrogens on this scale. So if I have an acidic solution, it releases lots of hydrogens, but do you see how the pH number is low? So the pH number is actually going to be low, anything from 7 to or 6.9 to 0. For basic, it's going to be anything from 7.1 all the way up to 14. This will become important when we talk about respiration and kidney function in 202. The acid-base homeostasis is really important to make sure that your blood stays at the correct pH. We'll come back and talk about this, but your blood pH should be around 735 to 745 for blood pH. We have lots of chemicals and nutrients and proteins floating around in our blood. They're going to various parts within our body, and they require the pH to be very neutral, or in at least this neutral range. So a slight change in pH can actually be fatal. So pH can actually break down and destroy some of the molecules that are floating around in our blood and make them inactive. And pH can be regulated by your kidneys, by your lungs, by chemical buffers. A little more detail than I want to get into right now, but again in 202 we'll come back to this. I want you to be familiar with the term buffer and its definition. It's a mixture of compounds that resist pH changes. We need to look at two different reactions that can occur. I just want to go over this briefly. I just want you to know the name and the definition. We will talk about this in much greater detail in 201 and 202. So if the pH rises, so think of pH rises, that means the number is high, right? So above 7.1 to 14. If the pH is between 7.1 and 14, we call this basic. What does basic mean? It means you have lots of OH ions being released. And if you have lots of OH ions being released, that means you have less hydrogens being released. So the pH is rising. So if the pH is rising, this molecule here, saying if the pH is rising, we're saying we have tons of these, right? Tons of hydroxide ions. So this molecule will actually split, and you'll release hydrogen protons. The hydrogen proton combined with the hydroxide ions, and you can actually form water. That will cause the OHs to go down. Why? Because they get trapped in water. So what's the opposite of this? If the pH falls, so when you think of pH falling, so pH falling should tell you an increase in hydrogen ions. So lots of hydrogen ions. What are we talking about? We talk about pH number. We're talking about 0 to 6.9, right? If I have lots of hydrogen ions, I want to do something about that. Because remember, any sort of pH change can cause issues with the chemicals that are floating around in your bloodstream. So if I have too many hydrogen protons floating around, it can combine with this molecule and actually create this molecule, this carbonic acid. And if I do that, if I've removed these hydrogen ions, I've taken them and I've sequestered them or hidden them into this compound, that means the hydrogen ion concentration is going to go down. So if the hydrogen ion concentration goes down, that means my pH is going to go back up to normal. I understand that this is difficult. It is. I just want you to start being familiar with it so when it comes up later, it won't seem so bad. Now we get to move on to organic compounds. So remember, organic compounds are anything that contain carbon. Usually we're also going to see some hydrogen, and there are a few exceptions. So carbon dioxide is not an organic compound, neither is carbon monoxide. These are unique to living systems, and then these are the four categories that we're going to talk about. So lipids are the same thing as fats. So we're going to go through each one of these in a little bit more detail. So before we talk about organic compounds, we have to talk about how we build them up and break them down. Sometimes we're going to want to store things like glucose, right? We may eat a sandwich, but we're not going to use the glucose right then and there to make ATP. So we have to have a way to store it. So you'll see the word monomer and polymer. A monomer, if I wanted to take monomer 1 and monomer 2 and put them together, that would create something called a polymer. We can also do this equation backwards. If I had a polymer and I want to break it up into individual monomers, I can do that too. And these are the ways that we do it: dehydration, synthesis, and hydrolysis. So dehydration, this is water, remove water. and synthesis is to make. So I'm going to make something by removing water. What I'm going to do is remove water from these monomers, and that'll cause a reaction to proceed that ends in a polymer. So these two get sandwiched together once water gets taken out. Hydrolysis means, hydro means water, and lysis means to break. So if I have a polymer, I'm going to hydrolyse with water. I'm going to use water to break the polymer apart into two separate monomers. Here's the more complicated reactions. They're showing you dehydration synthesis. Here's monomer 1, here's monomer 2. You can see the water molecule is going to come out, and then the two monomers are now linked by a covalent bond. So this would be dehydration synthesis. I'm removing water and I'm synthesizing or making something. In hydrolysis, I'm going to split. So now here's the polymer that I just created. Here's the polymer. I'm going to add water right here, and that's going to split the polymer into monomer 1 and monomer 2. And it's obviously more complicated than that. They're showing you just little blocks. This is what it would look like if we had the molecule and we have all of the elements making up that particular molecule. So I usually bring this up because I want us to understand why dehydration synthesis and hydrolysis are very important in human anatomy. So let's say you eat a big sandwich. So you've got tons of glucose here, but you've decided you're going to go to bed. You're not going to do anything. We want to take these glucose molecules and we want to store them. And we store them as something called glycogen. And we can store this in the liver. Then you wake up the next morning and you decide you're going to go for a run. So we take the glycogen and now we use water to break it down into individual glucoses. Remember, I have to have individual glucose plus oxygen to get energy out. So the importance here of dehydration and hydrolysis, the whole reasoning, is that to be able to store nutrients to use later. If we didn't have dehydration, synthesis, and hydrolysis, we'd have to eat every time we wanted to generate energy, and that would be kind of a pain, right? So this is a great way to take monomers like glucose, store them for later use, then pull them out of storage and break them down and use them for an immediate energy source. So let's look at carbohydrates. Basically, this is sugars and starches. We keep saying glucose over and over again, so that's probably the one that we think of. And here is the standard molecular formula. For glucose, it's C6H12O6. And then we can break carbohydrates into monosaccharides, disaccharides, and polysaccharides. Pretty simple prefixes there. Mono means one, di means two, poly means many. So carbohydrates are going to be our immediate fuel source, our major fuel source. So most muscles and nerves need glucose. They don't really want fat. They'll use fat if they have to. Well, muscles will, but they don't want it. They want glucose to make ATP. You'll also see carbohydrates, specifically sugars, that make up ribose and deoxyribose, which make up our RNA and DNA. Now, the nucleic acids, one of the other four groups we'll talk about in just a moment. So here's some examples: monosaccharides, disaccharides. You can see that we've taken two monosaccharides and made a polymer, right? We still know these terms, monomer and polymer. So we took monomer 1 and we took monomer 2 and we put them together, linked them together through hydration synthesis, yes, and created a polymer, but we're going to specifically call the polymer a disaccharide. The last category here is a polysaccharide. I just wanted to show it to you. Here's that glycogen I was talking about. So what is glycogen? You take a bunch of individual glucoses and you link them together. This is how we store glucose long term in the body. There are some other types of polysaccharides in nature, but glycogen is the one in the human body, so that's what we're going to focus on. Lipids are next. Lipids are basically fats. So lipids are going to be insoluble in water. This is just like if you put oil in water, right? The oil separates. There are major types that we have to look at. There's triglycerides, which means There's three. We'll see these in just a second. 3 fatty acid tails. We have phospholipids, which have 2 tails. And then we have steroids, which are kind of a ring complex. So basically, it's just a structural rearrangement of a lipid. Triglycerides, this is one type of lipid that we have to talk about. There are 3 fatty acid tails. They can be constructed of saturated fats or unsaturated fats. I have a figure in just a second to show you. But this is the most important part of the slide. What are the main functions? So triglycerides can serve as a secondary energy storage. So if you run out of glucose, you can break down fat and you can use that to supply energy to the body. Insulation, we usually see fat as a cushion or to trap heat in. So protection and insulation are also 2 roles. So let's look at the next slide and look at saturated versus unsaturated fats. So this is an example of a saturated where every single carbon has a hydrogen attached to it. You can see here this is unsaturated because these 2 carbons are missing hydrogens. So what's interesting is that the saturated usually stacks up, the fatty acid tails stack up, so this is usually a solid at room temperature. Unsaturated instead have kind of a kink in their tail, or tails plural, and so these are usually a liquid at room temperature. So think oils. For saturated, think butter. All the things that are good for you are unsaturated. All the things that taste good are usually saturated. The second group I want to talk about are called phospholipids. These are modified triglycerides. Instead of having 3 tails, they have only 2 fatty acid tails. Basically, they're going to have a head region and they're going to have a tail region. So head region and 2 little tails. The head is the polar region. This likes water. And the tails down here are repelled by water. So these are the fatty acid tails. And we know that fats or lipids and water don't mix. So the heads like water, and the tails hate water. They also use the terms hydrophilic and hydrophobic. This is important in cell membrane structure. We talked about having a border, right? Maintaining a boundary or border. So if we have a cell, this outer circle is called the plasma membrane, and it separates the external environment from the internal environment. Here's a typical structure of a phospholipid. So you can see the head here, and you can see the tails here. And you can see that there's only 2 fatty acid tails instead of having 3. So it's really just a modified triglyceride. The third category of lipid to discuss are steroids. Steroids have these 4 interlocking ring structures. Some common steroids, we think of steroid hormones, vitamin D that we'll talk about in integumentary, cholesterol. Cholesterol is really important in the cell membrane structure, the plasma membrane. When we get to integumentary, we'll talk about vitamin D, which helps us absorb calcium into our bloodstream, and that vitamin D is actually a steroid structure. Here's a figure from your book so you can see the 4 interlocking rings. Just keep in mind that a steroid is a modified lipid. So before we move on, let's go ahead and look at the plasma membrane structure. So here is the head of the phospholipid. Here are the 2 fatty acid tails. Then over here there's another layer where there's a head and 2 fatty acid tails. We call this a phospholipid bilayer. So you can see right down the middle here that there's 1 and 2 layers. Remember that the tails hate water. So the heads go towards the water and the tails away. So naturally, this bilayer is created where the tails are trying to get as far away from water as possible. So we can also see here these little items, these little green circles, these are called tags. So these are glucose or carbohydrates. I'm going to abbreviate. Carbohydrate tags. Sometimes we call these identity markers. So here it says carbohydrate. Carbohydrates serve as identity tags to let your body know which cells belong to you. So if there's any foreign invader, like a bacteria or a virus body, your immune system can tag it and fight it because it knows it doesn't belong there. So the first category we talked about were carbohydrates, and then we talked about lipids. The next that we're going to talk about is called proteins. And I wanted to show you proteins before we start talking about them. These purple blobs are proteins. You see that there are some proteins embedded in the plasma membrane. There are some other proteins that will be inside the cell, and then there's some proteins that the cell will make and ship outside of the cell. So let's talk about proteins. Proteins are the third category of organic molecules that we're going to talk about, and they have the most varied function. So they can serve a structural purpose like we saw in the plasma membrane. They can serve an enzymatic or chemical function. They can make reactions happen or increase. We'll also see them when we talk about muscle contraction. We have proteins, myosin and actin, that need to come in contact with each other for a muscle to contract. They all contain the four major elements, the carbon, the hydrogen, oxygen, and nitrogen. All proteins are made from 20 types of amino acids, and these are the monomers. Remember, we would say if we had monomer 1 and monomer 2, put them together, and you have a polymer. Well, in this case, the polymer would be called a protein, and the monomers would be called amino acids. So not only does the order of amino acids matter, but also the shape. So if you have amino acid 1 and amino acid 2, or if you have amino acid 3 and amino acid 4, these are going to give you two completely different proteins. And I'll show you this on the next slide. But the shape also plays an important role. So how these amino acids link together decides how the protein is going to function in the body. So here's the beads on the string, if you will. Here's amino acid 1, 2, 3, 4, and 5. We've linked them together on a string. We call this the primary sequence. Again, I want to point out, if I had amino acid 2, if I shuffled these, if I had 2, 3, 1, 5, and 4, this one would give me protein A. This arrangement would give me a completely different protein. This would be protein X. So the amino acid sequence matters, and then the shape that the amino acid sequence folds into matters as well. So here are examples of secondary shapes. So once we link the amino acids like beads on a string, then we can have interactions between different amino acids depending on their location. So we call this a ribbon. It's really called an alpha helix. And then we have beta sheets. So you can see that there are amino acids here, but they're actually interacting with the amino acids that are next to them. Third kind is called tertiary. So you can see we have beads on a string here. This is primary amino acids. Then we have an alpha helix, and then we have a beta sheet. So there's 3 items inside this tertiary structure. We have the primary, and we have the secondary structure. The fourth protein structure is called quaternary, and quaternary, you can see here, it's really just 2 tertiary structures put together. So enzymes are a very specific type of protein. We say they're proteins with a specific job. So enzymes are the ones that are going to go out and cause chemical reactions to increase. So this is a protein that acts as a biological catalyst. They give a fancy definition. It says lowers the energy needed to activate a reaction and increase the speed of the reaction. Basically what's happening, if we say it's a biological catalyst, it speeds up the reaction time without being used. So what's important about that is that if you have an enzyme, it can do the same job over and over again. It doesn't get used up in the reaction. They're usually named after the reaction that they catalyze. So in terms of the example here, it says sucrase. So sucrose is a sugar, right? It ends in OSE. It's a sugar. It's a disaccharide. And the enzyme that breaks sucrose up into its original components, which are glucose and fructose, is called sucrase. So here's an example. Here's the enzyme. Now remember, enzymes are proteins. And what did I say about shape? The shape of a protein is important, right? You can see why the shape is important on this enzyme because there's two active sites here, and these molecules, we call them a substrate if it's combined an enzyme. So this would be, substrate is the same thing as a reactant, but we just know it's specifically a substrate means it's going to bind to an enzyme. So a substrate is a reactant that will bind to an enzyme. So in this case, let's say we have a glucose and a fructose, and we want to put them together to make sucrose. So the glucose and the fructose bind, and when they bind the enzyme actually hugs these two molecules and allows for this bond to occur. And remember, water is going to be released. What was this called? Dehydration synthesis, right? So if I want to take two molecules and put them together, I'm going to pull water out, and now I'm going to have a polymer. Enzymes are responsible for aiding these reactions and making them happen more quickly. Here's where pH comes in. So we talked a little bit earlier about pH. If a protein loses its shape, so remember that other picture, the protein kind of looked like this, and it had those two little spots where the active sites. What happens if the protein changes shape and now it looks like this? There's no way that those molecules can bind the enzyme anymore. So we call this protein denaturation. So if a protein loses its three-dimensional shape or structure, it can no longer carry out its task or function. And a great example of that is this chicken egg. So this white stuff here was going to be, I call it baby chicken food. What was this going to be? That was going to be the baby. And this was going to be the food that was going to feed the baby until the egg hatched. So what do? We crack open the egg and we fry it up. Temperature, increased temperature, can cause denaturation. So when you fry the egg, you can't ever turn the egg back into the baby or the baby chicken food. Why do we have to study enzymes? Why are they so important? Not only do they carry out tasks within the body, they also carry out photosynthesis. So go back to that moose picture that I showed you where the sun is shining down on the tree and the tree creates glucose, and then the moose eats the tree leaves and is able to do mechanical work. So the photosynthesis basically turns the sun into chemical energy. What was that chemical energy? It was glucose. Then we eat glucose. We eat plant matter, right? So we consume the glucose, and during cellular respiration, we turn glucose into ATP, and this gives us the energy that we need to carry out tasks within the body. So both of these reactions are necessary for us to generate energy, and both of these require very specific enzymes in order to occur. So basically, if anything happens to the enzymes responsible for cellular respiration, you die, right? So if you don't have ATP, your muscles stop working, your brain stops working, so you need ATP, you need cellular respiration, and cellular respiration requires enzymes to occur. If you want to see a nice picture from another book, it just kind of shows you, the cycle of life, so to speak. So here's the sun. The plants take the sun and they turn it into glucose. We take glucose and consume it along with oxygen, and then we turn it into carbon dioxide and water. Remember, the carbon dioxide we exhale, and the oxygen, how did we get it in? We inhaled. So cellular respiration and photosynthesis are linked. You can't have one without the other unless we can find a way to generate an alternative food source without using plants or animals. So here's our big critical thinking question. If enzymes are necessary for cellular respiration, and cellular respiration generates ATP, basically energy, mechanical energy for us, What happens to us if the cellular respiration enzymes are denatured? I don't know how many ways I can stress this, but you die. So why is a fever, if you get a really high fever, why is that dangerous? If you have increased temp, it can cause pH changes. pH changes can cause enzymes specifically to do what? Denature. Nucleic acids are the fourth category that we have to talk about. So nucleic acids are the largest molecules in the body. We have two major classes. We call them DNA and RNA for short, but they're really deoxyribonucleic acid and ribonucleic acid. The monomers here are called nucleotides. So just like we said the amino acids were the monomers and the protein was the polymer, now we have the monomers are called nucleotides, and nucleic acids would be the polymer. So you link a bunch of nucleotides together, you get a nucleic acid. The first type of nucleic acid, deoxyribonucleic acid, DNA for short, it has 4 bases, A, T, C, and G. It's double-stranded, and basically DNA provides the instruction code for protein synthesis. So DNA eventually is going to code for proteins, all the proteins that we make in the body. And remember, we said these have various functions. We can do muscle contraction because of proteins. We have structural proteins. We have enzymes that are proteins. This is a great picture to get an idea of what a nucleotide consists of. So every nucleotide has a phosphate, a sugar, and a base. And you can see that the two nucleotides come together. They're held together by hydrogen bonds. Remember hydrogen bonds? And that's interaction between positive and negative forces, right? And it forms this ribbon structure and called a double helix. There's 2 strings that are intertwined, held together by hydrogen bonds. Ribonucleic acid is the second type of nucleic acid. It has 4 bases as well, but you'll notice the one in red, uracil, it's different than the T that's in DNA, right? So 3 of the bases are the same, but 1 is different. RNA is also usually single-stranded, and RNA carries out the orders for protein synthesis from DNA. So basically, DNA copies orders to RNA, and then RNA makes protein production happen. So without RNA, you can't make a protein. The DNA stays trapped inside the nucleus, and we'll talk more about cell structure in chapter 3. But there are different types of RNA that are going to play different roles in the conversion of DNA information into a protein.
DNA and RNA are nucleic acids, polymers made of subunits called nucleotides. One difference between DNA and RNA is the type of sugar their nucleotides contain. DNA contains the sugar deoxyribose, while RNA contains the sugar ribose. Ribose has 1 more oxygen atom than deoxyribose. DNA and RNA are each composed of 4 different nucleotides, which differ in their nitrogenous bases. 3 of the 4 bases are the same in DNA and RNA: adenine, guanine, and cytosine. The 4th base in DNA is thymine. In RNA, it is uracil. The nitrogenous bases guanine and adenine each have 2 linked rings of atoms. They are called purines. Cytosine, thymine, and uracil each have a single ring, and these 3 bases are called pyrimidines. For convenience, the carbon atoms in a nucleotide sugars are numbered, beginning with the carbon atom bonded to the nitrogenous base, moving around the ring, and up to the carbon that is bonded to the phosphate group. The 1' carbon is bonded to the nitrogenous base, the 3' carbon to the next nucleotide, and the 5' carbon to the phosphate group. DNA and RNA are polynucleotides, long chains of nucleotides. Polynucleotides are always assembled in the 5' to 3' direction. A covalent bond forms between the carbon at the 3' position of a nucleotide and the phosphate group at the 5' position of the next nucleotide. RNA usually consists of a single polynucleotide chain. DNA, on the other hand, consists of 2 polynucleotide chains. The two DNA chains, or strands, are oriented in opposite directions and held together by hydrogen bonds between the nitrogenous bases on opposite strands. Because of their sizes, shapes, and arrangement of polar groups, the DNA bases form complementary pairs. Adenine pairs with thymine, and cytosine pairs with guanine. The two polynucleotides in DNA wind around each other to form the familiar double helix. The bases can be in any order, like the letters of the alphabet. However, the base sequence is significant. Sequences of bases called genes encode the instructions for the structure and function of an organism.
Adenosine triphosphate is the third type. Adenosine triphosphate is a nucleic acid, but it's also what we called an energy storage, right? ATP is the energy currency used by the cell. So basically, we take glucose, we break the bonds, we store energy in ATP, and then ATP can go other places within the body and release the energy. So at the very basic level, it's very similar structure-wise to RNA and DNA. The breaking of high energy bonds releases energy that the cell can use to do work. Right here, these pink little squiggly lines, they're trying to show you that that's where the energy is trapped. The energy that was in glucose gets transferred and is trapped in these bonds. So here's the adenosine. It has a sugar, it has a base, and it has a phosphate group. So this is a nucleotide, right? A sugar, a base, and a phosphate. If you have only one phosphate, you're monophosphate. If you have two, if you have three. So in these bonds, we have lots of energy, stored energy, high energy phosphate bonds. If you break that bond, energy is released. So what's going to happen is this ATP molecule is going to go somewhere in the body, and we're going to break this bond between the phosphate. We're going to release tons of energy, which can allow the body to do work, and we're going to be left with ADP. This is 2, right? ADP, plus we'll have a P, but we'll be missing some energy. The energy was released in order for mechanical work to happen. The interesting thing about ATP, we can see on the next slide. ATP has a cycle, or some people say it's cyclical. So here's my triphosphate. If I break this third bond between the second and third phosphate, I'm going to get tons of energy out. I still have ADP left plus my phosphate. So the only thing that came out was the energy. Then ADP plus P, if I put energy in, that will reform ATP, and then I can use ATP again over and over and over again as an energy storage molecule. So how do we use ATP in the body? We're going to use it in numerous ways. Some we'll see in more detail than others. But here's a protein, and this protein is trying to pump solutes. Solute, it could be salt, it could be calciums, it could be potassium, but this protein is trying to pump a molecule from one side of a membrane to another. ATP binds and gives the protein energy so that it can change shape and dump the solute on the opposite side. We'll see mechanical work. We'll see how ATP actually causes muscles to contract. And then we can see chemical work. This is basically, they're showing you an example of an enzyme, right? So what can ATP do? It can cause two monomers to be joined or to create a chemical bond between two monomers and create a polymer. Well, that's it for Chapter 2. Hopefully it wasn't too painful, and I'll see you next time. Bye.